The best way to eliminate any bias or previous perceptions a scientist might have is to follow the scientific method. The scientific method is a tool that helps scientists and the rest of us to solve problems and determine answers to questions in a logical format. It provides step-by-step general directions to help us work through problems. Although different sources describe the steps in different ways, they fundamentally incorporate the same concepts and principles. For our purpose, we're going to utilize the key steps shown in the diagram to the right. There are 5 steps to the scientific method. Make observations and identify a problem. Ask questions and research the problem. Formulate a hypothesis. Conduct a controlled experiment, and reach a conclusion. Almost all scientific inquiry begins with making observations. Our observations cause us to wonder, to ask questions, and to do research. Through questioning and research, a scientist can narrow down the focus of their inquiry by identifying the problem in specific terms. In an informal lab report, this information becomes the purpose of the lab. In a formal lab report, this information becomes the introduction to the paper. The great thing about a question is that it yearns for an answer. The next step in the scientific method is to suggest a possible answer in the form of a hypothesis. A hypothesis is often called an educated guess because it almost always is formulated from research and what you already know about the topic. When formulating the hypothesis, there are 2 important qualities it must have. It must be testable and falsifiable. A testable hypothesis allows a scientist to determine how 2 variables are related. A falsifiable hypothesis allows a scientist to determine that a hypothesis is false if the observations are true. For example, when examining the corn plants shown in the picture to the left, you notice that the plants grown after a soybean crop are taller than the plants grown after a crop of corn. Why? After doing some research, you learn that the soybeans act like a fertilizer. Using this information, the hypothesis is formulated. Corn with fertilizer will grow taller than corn without fertilizer. The hypothesis is testable and falsifiable. Now it's time to develop the experiment. This step in the scientific method requires the scientist to develop a controlled experiment to test the hypothesis. A controlled experiment sets it up so that a control group and an experimental group do exist. The control group receives normal conditions, whereas the experimental group receives the variable to be tested. The control group allows the scientist to compare his results against a baseline measurement so he can feel confident that the results are not due to chance. Controlling an experiment also means controlling the variables so that only a single variable is studied. All variables other than the one to be tested must be held constant for both the control group and the experimental group. The independent variable, also called the manipulated variable, is the variable to be tested and is manipulated by the scientist. As the independent variable is changed, the dependent variable, also called the responding variable, is measured for any variation. In our corn example, the independent variable is the fertilizer and the dependent variable is the height of the plant. During the experiment, scientists collect both qualitative and quantitative data. Qualitative information is based on the scientist's senses and is described in words in what is being observed, whereas quantitative information is acquired by measurement instruments such as rulers, balances, graduated cylinders, and others. For example, the height of the plant can be measured with a metric ruler. Buried in the qualitative and quantitative information, hopefully, is the evidence to support or reject the hypothesis. The amount of analysis required to come to a satisfactory conclusion can vary. Sometimes a simple analysis of the qualitative data can be done, and other times a statistical analysis is required. However, either way, the goal is to support or reject the hypothesis. Once observations have been made and data collected, the process of communicating our findings gets underway by making a conclusion. A conclusion should explain the data, communicate if the data supports or refutes the hypothesis, contain an error analysis, and describe how the experiment can be improved to eliminate such errors. Remember, the goal of all science is to explain and predict natural phenomena. So what comes next? A generalization that describes a behavior in nature is called a scientific law. Laws may be expressed as mathematical equation or concise statements. They describe or predict, but do not explain natural behavior. Once a successful prediction can be made in an area of science, it becomes possible to move on to the task of explaining why something occurs. To do so, scientists often engage in constructing models. Models are visual, verbal, and mathematical models that show a relationship amongst information. In many cases, a model that is successfully applied may become part of a scientific theory. A scientific theory is a well-tested hypothesis that explains a body of known facts or phenomena. Theories can never be proven, but they are considered successful if they can predict the results of many new experiments. Throughout every stage of the scientific method, testing takes place continuously. Scientists test data and results of experiments by carrying out new experiments. No hypothesis, law, or theory is ever free from testing. All are constantly open to modification or abandonment based on new evidence. Science is a method. It's not what you study, but how you study it.